Our first presenter is Jonathon Borrelli, and he's one of the many researchers associated with the Jefferson Project. As Kathy referred to, Lake George is the smartest lake in the world, and that's due to the work of our partnership with IBM and RPI. John has numerous publications and applied network theory to answer ecological questions about community structure and dynamics. But he also has a project called the Dashing Gecko, which was interesting to find out, where you can actually purchase stickers of artwork of the various aquatic life that he has worked on and researched. So that's pretty interesting. But this morning, John will be presenting on the near and offshore chloride data that's presented from the Jefferson Project. So please welcome Dr. John Borrelli.
Thanks, everyone. I see you visited my website. Thank you for that. So I'm a postdoctoral researcher with the Jefferson Project. Mostly I work on modeling the food web of Lake George, trying to sort of understand how different organisms interact with each other and how those interactions affect their long-term dynamics. But since this is the Road Salt Summit, and I work a lot with our survey data, I thought I'd take some time to present a little bit about chloride in the lake and what we found regarding that. So just to start off with, in case you aren't very familiar with the Jefferson Project, we are, as was mentioned, a collaboration between Rensselaer Polytechnic Institute, IBM Research, and the Lake George Association. And our goal is to use science and technology to identify human impacts, suggest solutions, and measure mitigation success. To me, the Jefferson Project really has, we're trying to answer three really big overarching questions using Lake George as our focus. First of all, we want to know, is the lake changing, right? What changes are occurring in the water quality, in the biotic environment, and as part of that, really, how are the human impacts on the ecosystem being realized in the lake? Given that the lake is changing, we also want to know how can we best explain those changes in the lake? In other words, we're looking for mechanisms, causes, why are these changes happening? And the reason we want to do that is once we have our mechanisms, once we understand why things are changing, we can use that to make predictions about the future. We want to be able to say, okay, if the environment changes in this way going forward over the next 10, 20 years, what is going to happen to the water quality of the lake? And so for most of this presentation, I'm going to be focused on this first question, is the lake changing? And to get at that, at the Jefferson Project, we've got a couple of survey tools that we have, data sets to help us address whether or not the lake is changing. In fact, there's really four data sets. The first one is an offshore data set, which was started in 1980 and is going all the way to today. Started off with 6 deep water sites and moved on to 11. They're in the open circles on the map there, pretty well spread out throughout the lake. And at those sites, we measure chemistry in the upper mixed layer and down near the bottom of the lake. And we also quantify biotic variables like phytoplankton and zooplankton. Our second survey started a little bit more recently. It was a nearshore survey, started in 2017, going till now. And there we sort of expanded on earlier work with the offshore survey. And moved towards having a sort of a really widespread, a lot of sites throughout the lake. Those are the black dots that you see on the map there. 30 sites throughout the lake, and there we measure water chemistry as well as biotic variables like zooplankton. We also measure macroinvertebrates, the little insects that are living in the lake. There we go. So I want to start off just talking about some work we did, published recently, on some of the long-term changes in the lake. using that offshore monitoring program, specifically focused on the upper mixed layer of the water, sort of that open water area, what's going on in there. We've measured 18 different water quality variables, and we're using 7 deep water sites with the longest time record. What we've learned, sulfate and nitrogen are decreasing. The sulfate is largely because of the reduction in acid rain. So that's kind of a cool result. Secchi depth, which is the water clarity, phosphorus, dissolved oxygen, and silica haven't really changed very much over the last 40 years. Alkalinity, chloride, sodium, and conductivity. So you might be particularly interested here in chloride and sodium and conductivity. They're all somewhat related, but those have been increasing the most. They have the highest increase over the last 40 years. But we're also seeing increases in temperature, chlorophyll, and orthophosphate, which is a form of phosphorus that is readily available to things like phytoplankton. Those have also increased, but not quite as much. If this is something that interests you, this is where I have my plug for our data dashboard that's currently up and running. You can find it at jeffersonproject.live. You can scan that QR code, it'll take you right there. But on that dashboard, you'll find information about the Jefferson Project and what we do. You will also be able to look at real-time sensor information from our many, many sensors active throughout the lake. You can look at hyper-localized weather forecasts. The guys at IBM are really proud of this. You can get very high-resolution weather forecasts pretty much all around the lake. Finally, you can look at some of the long-term water quality trends that I've mentioned here. Basically, just take a look and see, for all these variables, what does their change over time look like on an annual basis. And as part of that, sort of brings me to what I want to mainly talk about today, which is chloride. And I want to do a little bit more of a deep dive into how chloride is changing in Lake George. So if you've been here before, if you're familiar with the Jefferson Project or the LGA, you might have seen a graph that looks like this before. Just to orient you, because you're going to see a lot of these graphs, on the y-axis is chloride in milligrams per liter, and on the x-axis is time. So here we have chloride from all of our offshore epilimnetic samples. That's the open water near the surface, top 10 meters of the water. And what you'll notice is chloride has largely increased, as I mentioned, going from about 6 milligrams per liter to around 19 or 20 milligrams per liter today. With some interesting features in that I'll discuss in just a bit. We also have data from the hypolimnion, which is near the bottom of the lake. So here you can sort of see a difference between sort of that upper layer and the bottom layer of the lake to see whether that chloride is pretty evenly distributed. And actually, you see pretty much the same pattern, almost exactly the same, really. Excuse me. With just a little bit more spread in the data. Now, we also have shallow water samples. We have 4 shallow water samples from 1980 on in association with that offshore survey. And again, we're seeing largely the same pattern that we see in the deep water samples, with just a little bit more spread there as well. And finally, for our nearshore survey, which hasn't been going on as long, chloride is roughly the same, at the same level as we see in our offshore surveys, with some outlier exceptions that I will talk about in just a bit. So on average, what we can learn from these different data sets is that on average, chloride is higher in the shallow nearshore areas than it is in the deeper water areas, but not by much. We're talking like maybe half a milligram per liter to 1 milligram per liter higher on average. But it is a lot more variable in the shallow nearshore areas. Excuse me. Here you're seeing the coefficient of variation, which is a measure of the spread of the data. In the light color are the nearshore areas, and the darker colors, that lighter color is that shallow water sample, and the green and blue are the offshore samples. And basically what that's telling me is that because that nearshore area is more variable, that's why we're seeing slightly higher concentrations there, because on average you see higher highs in the nearshore area than you see in the offshore regions. So one of the other things we can do is we can look at how exactly has chloride been changing, like what is the rate of change? And to get at that, I fit a function using something called a generalized additive model, which basically just accounts for wiggliness in the data. And I use that to determine at each point in time how fast is chloride changing. And what you see over here on the right-hand side, on the right-hand graph, is that rate of change over time. And for most of that period, it's increasing. But it's not increasing at the same rate. Sometimes it increases a little faster, sometimes it increases a little slower. What's interesting is this period here. So starting in about 2010, where you see that gray line and red line, chloride is actually leveling off, actually declining a little bit. But then in 2015, and you can actually see this on the time series here, right around 2015, there's a jump. It almost kind of looks like a break if you squint at it. There's that jump in chloride, and that's what you're seeing in this big peak on the right-hand side of the graph. So something happened in 2015, or something happened in 2014 to cause that initial decline and then big jump. I'm not entirely sure what it is, but then starting again in 2020, that's when we start to see it leveling off again. So largely, that chloride is kind of leveling off. Using a similar approach, I was looking at chloride in space. And for this, I used a method to interpolate our nearshore and offshore data. Pardon me. And what we can learn from this is that on average, chloride is higher in the south. But I will note that the gradient is not very big. It's about a 4 milligram per liter difference between the south and the north, which might be statistically significant, but is not necessarily biologically significant. And finally, I wanted to put chloride in a little bit of context. So here you'll see a graph with two of the lowest water quality thresholds for chloride, Canada and the US EPA. Canada guideline is 120 milligrams per liter. US EPA is 230. And so overall, Lake George is far below the lowest thresholds for aquatic life, which is good. We do have one sample, which you might have noticed from that nearshore graph, that does come close, and that is a sample from East Brook in June of 2019 at 85 milligrams per liter in the lake. We have never seen anything as high as that before, and we haven't seen anything like that since. I haven't had a chance to look through our meteorological data or our stream data. I can't explain that outlier just yet, but it could be a result of some strange weather event sort of flushing a lot of chloride out of a stream. But our second highest sample was about 35 milligrams per liter, and that's from Lake George Village in March of 2006, just to give a little bit of context there. So some of the takeaways for chloride in Lake George. We do have higher and more variable concentrations in the nearshore. We have higher chloride in the south than in the north. And then except for a jump in 2015, chloride has largely been level since 2010. So probably doing a good job, guys. Just going to finish up a little bit, talking a little bit about these second questions, and then I'll finish up. This is where my bread and butter is as a researcher, thinking about how we can explain changes in the lake and making predictions about the future. And for that, I turn to food webs. Food webs are models of who's eating whom in the lake, how the species are interacting with each other. And the reason I like using them is because they provide a context for how changes in environmental variables like chloride or nutrients or temperature, how those environmental changes propagate throughout the community and have larger impacts. We get a lot of our information from running experiments, so I wanted to just highlight this experiment from a former postdoc with the Jefferson Project who ran multiple experiments in these mesocosms. They're large cattle tanks, 200 liters or so. And they measured change in zooplankton and chlorophyll, which is a proxy for phytoplankton, after 1 or 2 months. And they tested a range of chloride from 0 to 1,500 milligrams per liter because they wanted to know sort of how does that increase in chloride impact the ecological community, how big of a difference does it make, and importantly, how good are those thresholds, that 120 milligrams per liter. And largely what they found is as you increase chloride, you see a decline in zooplankton, which are important micrograzers in the food web. They eat down the phytoplankton. And so when you lose those zooplankton, you see that green line increasing. That's an increase in phytoplankton abundance. More importantly, the concentration at which 50% of the population of zooplankton was lost, that's a sort of an important measure. It's called the LC50. What they found is that it's often close to the lowest thresholds, that 120 milligrams per liter. It's sometimes below that threshold. So whether those thresholds are really great for protecting water quality is an open question still. Other things we've learned from experiments, the species present in the experiment can influence the outcome, whether predators are there or competitors. The experimental design can lead to differing results, whether you're using cattle tanks or in-lake mesocosms. And then if you have multiple stressors, increasing temperature, changing light, that can also interactively impact species. So in my work, I look at the Lake George food web, which is actually fairly large and fairly complex. This is actually a somewhat of a simplification of the Lake George food web network. It's a map of all the interactions between phytoplankton, zooplankton, macroinvertebrates, fish, plants. And there's about 180 species in there with about 1,400 interactions among them. Well, what I can gain from this, I've been able to use this to sort of build a model for how the dynamics of these species go. And what I get from that, I can actually do a fairly good job of replicating the observed dynamics. So here we have 3 different kinds of zooplankton. The black dots are our observations. The red line is what my model says their abundance should be. And you can see that the model does a pretty good job of replicating that. And the reason that's important is because once we can replicate the dynamics, now I can start messing with it. I can say, okay, what if we increase the temperature? What if we increase the chloride? How's that going to change things? And so that's going to be one of our future goals. And so just to finish up, salt has increased in Lake George over 40 years, but it seems to be flattening. That curve seems to be flattening. The impact of salt on species in the lake depends on the presence of competitors, predators, and other stressors. And we are working on incorporating experimental data into our models to make our predictions about long-term impacts of both increased chloride and other stressors. And with that, I'll take any questions.
Jonathan, that one chart you showed where there was a large swing, 2010, 2015, big curves.
Mm-hmm.
Did you look at the amount of snowfall in those years to see if that may have played a part? Because snowfall usually means, we do a good job here with it, but it usually means that there's more salt in the roads.
That is a great question. I have not had a chance to look at that yet, but that is definitely the first place I'm going to look. There's a couple of explanations. It could be, maybe 2013, 2014 was a bit of a milder winter. Maybe you used less salt, so maybe there was kind of a decline, a little bit of flushing, and so there was a kind of a downturn, and then it kind of normalized after 2015. Or it could be that winter of 2015 was especially harsh, and maybe you had to use more salt, so it kind of jumped up a little bit. I am not sure what the explanation is, but we are going to look into that. But yeah, very good question.
If I could. Oh.
Thanks, Bria. Thanks, Jonathan. That was helpful. One question, we talk about the lake and the deep water in the near shore and the higher levels in the near shore. What about the streams? Can you speak at all to the streams? Because our understanding is that's where we're seeing the real spikes, beyond thresholds that we're concerned about.
Yeah, another good question. So we do have stream data. I have not personally looked at the stream data very much, so I can't speak to that right now. I do know Joel gave a presentation on stream data last year, which is online if you want to check it out. But you're right, we do see a lot higher concentrations in those streams than we do in the lake. And that is something that is very interesting because, yeah, those high chlorides, they can influence other things that are coming into the lake as well. So yeah, we are on it.
I know. Thanks.
And if I could, John, sorry, as a follow-up to that, John, could that explain some of the variation, in the nearshore data that you were just showing that, there's higher concentrations nearshore, and that could be the influence from streams? Possibly.
Yes. So a lot of our, especially the more recent sites from 2017 on, a lot of those sites are at the stream outlets. And we don't really time our surveys based on weather, so there could have been surveys where we go out and immediately following a storm event or something like that, and that can strongly influence whether or not we're finding high or low chloride at those sites. Though I think it's actually might be a little less likely in that long-term data set because I think those shallow sites are not at stream outlets. But I'd have to double check that.
Yes. Any other questions? If not, thank you very much, John. Appreciate it.
Thank you.